But I want to start our ecology unit, which we're starting with this lab, and which we will pick up next week. um, with an advertisement, since we're still in the advising period, for the ecology class that runs in the fall. So if any of you are interested in ecology, if this topic excites you, as we start to talk about ecology, it's something you want to study more, we do offer a 200 level Foundations of Ecology class, and this class is required for environmental science, natural resource conservation majors, and it transfers well into the biology department, even at UMass. It will count as your prerequisite to upper level ecology classes. So if you're interested in ecology, this class is available. If you don't like my teaching style, I'm the only one that teaches it, so you can factor that in to whether or not you want to take it. You'd be stuck with me again. But a couple of people, the Taylor's going to show up here in a minute, and Riley can tell you about it if you want to know. We do spend a lot of time outside, so ecology is sort of the study of the relationship between organisms and their abiotic environment. So we're at the point where we're actually looking at the whole organism themselves, how organisms interact with each other, and then how organisms interact with their surrounding environment. Lots of time in the field. Um, so what I want to do today is just talk to you a little bit about population ecology. And so this is my sort of preamble to the population ecology lab. I use this slide not just to show you that I used to be young, but also because I've been studying population ecology or actively doing population ecology as a researcher since like my junior year as an undergrad. So for a really long time, and I know I put the dates on there, I'm totally telling you how old I am. Um, for a really long time, I've spent like a lot of my career studying population ecology. It was never because I was super interested in population ecology, but because population ecology is a really important background, like something you have to do in order to study anything else about the organism that you're interested in. And so I want to talk a little bit about what a population is and how we try to measure populations. And I'm going to do this lecture from a very animal-centric place because we're going to go capture animals and we're going to talk about the uniqueness of this method as it applies to animals. There are lots of ways that we get at the same questions for plants and fungus and other organisms, but things that don't move and hide are counted and quantified in different ways than things that can move and hide. So this is sort of based on animals who can escape your attempt to count them. What animals are those? This is Microtus pennsylvanicus, which is a meadow vole. This is Dipodomys miriami, a Merriam's kangaroo rat. This is a Dipodomys miriami, Merriam's kangaroo rat. This is a Paragnathus parvus, which is the Great Basin pocket mouse. And this is Ovis canadensis bighorn sheep. I guess I should make the bighorn sheep come forward because you can't see his little, here's his little face down here. I think he used to be forward and he went backwards. Need to rearrange the pictures. Nice ram. Okay, so in order to talk about populations, we have to first know what a population is, right? So what is a population? We can just loosely define a population as a group of individuals living in the same place at the same time, right? We are the population of biology 2 at GCC spring 2019, right? We are a subpopulation of students at Greenfield Community College. We are a subpopulation of people who live in the Pioneer Valley or people who live in southern Vermont, right? We're all have different scales at which we can think about populations. And then the second question, what is ecology? I kind of said this already. Ecology is just a study of the interrelatedness of organisms, both with each other and with their non-living environment. So how do members of the same species interact with each other? How do they interact with members of other species? And then how do they interact with the non-living environment, the soil, the temperature, the climate, the humidity, all of those other components that are essential to their lifestyle. So population ecology usually boils down to the study of the distribution and abundance of organisms. Basically, where do organisms live and why, how do they live there? And then how many of them are there in that place? So I have this picture to kind of illustrate some of the complexity of thinking about a population. I know it's not super clear for those of you who haven't been to this part of the world, but this is Wyoming. This is Montana, right? This is the greater Yellowstone ecosystem. This is Yellowstone National Park. And so we could look at this map and try to determine population size for something like the grizzly bear, right? And if you were a wildlife biologist working for one of the two affected states, or if you were a federal game biologist working for the federal government, you might use boundaries like state boundaries and county boundaries and national park boundaries to define your population, right? But if I'm interested in understanding grizzly bears in Yellowstone National Park, and I draw this box around Yellowstone National Park, and this red boundary here is actually suitable grizzly habitat, it's really irrational to pretend that these grizzly bears walk up to this imaginary line in the forest and go, oh, I can't leave the park, right? No, of course they come down here into the Shoshone National Forest. Of course they travel to Grand Teton, because why wouldn't you travel to Grand Teton? It's beautiful. Like, they're going to go throughout this whole area. And so often we lay these sort of artificial boundaries on things, our human interested boundaries. But if we really want to understand a population of animals, we can't just use our human boundaries. We have to use the natural boundaries of the species. Where are they capable of living? Where is the habitat suitable? Where is there enough food? Where is the right climate or the right elevation or the right interaction with food sources? And so we would want to actually look at this whole piece. For things like large mammals, like bears, that gets really complicated, because now we've got to get the National Park Service, the federal government, the state of Montana, and the state of Wyoming all to agree to let people do that work in that place, right? And so, but if you want to do population ecology well, you need to use natural boundaries, not artificial boundaries. Okay, what are the things that affect population size? This is pretty basic. There are demographic factors that affect population size, births and deaths, right? New individuals are born into a population and individuals leave a population when they die. This is a natural increase or decrease rate. And then we have geographic factors like the movement of individuals between populations. And so individuals might immigrate into a population or emigrate out, right? They might leave a population or join a population, and that gives us net migration. So we can think about a population changing due to birth and death, and a population changing due to immigration and emigration. So like right now, New England, you listen to anybody in higher ed is talking about the crisis of enrollment, right? The population of New England is declining. We have fewer births, and we're not catching up to the death rate with new births. But we also have not very much immigration relative to other parts of the country. And so we're not replacing that loss in births with immigrants from other places. And so the population as a whole is declining. So New England population is going down. It does make for nicer forests, right? Okay. So when we want to think about how big of a population is how big is a population, you might say, well, just count them all up, right? That should be easy. Just go out and count. No, it's actually virtually impossible. We're coming up on a new census for the United States, like the number of humans that live in the United States. If you're paying attention to the news at all, there's a big battle going on right now about a question on the census and whether or not people will choose to not fill out the census because of questions that are on it. Even if you have individuals who you know where they live and you go and ask how many people you live in your house, they might tell you not the truth, right? With animals, you go out and count them. Some of them might hide from you. some of them might be able to evade your ability to see them. And so, although a census sort of is the gold standard, a complete count of all individuals, that's what you really want, right? But it's difficult and rare, and in most cases, completely impossible. And so, we almost never can conduct a census. You can conduct a census if you have organisms in captivity. People have censused sections of the forest. I mean, these studies are terrible. I tell you guys all about these terrible studies. They've actually put plastic bags over trees and then bug bombed them and then counted like all the dead invertebrates that rain down from the trees. So that would be a way to conduct a census, right? But we can't do that on a global scale, obviously, for lots and lots of reasons. And we probably shouldn't do it on a tree by tree scale because of what the destruction that happens with that, right? So you can't shake the globe and get all the animals to fall out and count them in a little pile. So censuses are really not possible. Typically, when you try to do a census, you end up with what's known as MNKA, the minimum number known alive. So you get some number, like I saw 100 bison. I know there's 100 bison. There might be 300, but I know there's 100 because I counted them, right? And so you could fly over this valley and count how many brown spots there are in the valley and get the minimum number of bison in that picture. Okay. All right. So since we can't shake the globe and knock all the animals loose and count them in little piles, we have to rely on estimates. We can't get a census. We can't get a whole count. So we either get, a minimum number known alive, or we try to estimate the population. And the way we estimate populations is by sampling individuals. So we take a sample. We go out and count as many individuals in the population as we can. We do that multiple times over time. We develop as scientists and mathematicians some sort of detection probability, how likely we are to see those animals when they're there. And then we use that to estimate the true population size, n. So your sample, little n, is the number of individuals you count. Little p, detection probability, how likely you are to find them. And then capital N, an estimate of population size. I know I've told some of you about the first time I ever encountered a kangaroo rat. It was the first time I went to the field in the desert, and it was night, and we got out of the truck, and we had just started to walk out to the site, and this rat hops up to me, stops at the base of my feet, looks up at me, turns around and kicks sand until the toe of my boot is covered, and then hopped away. And that was this guy, a desert kangaroo rat species. And I worked on that plot for over a year before I ever captured a single desert kangaroo rat. And I would see them, and they would come and they'd hop and kick sand at me, and I would like watch them because they're really big and they're obvious. You can see them with your naked eye, but I would almost never capture them. So that species has a really low detection probability. They're hard to catch. They're hard to find. And so that makes your estimates have more play around them, right? Your estimates are not going to be as good as the species that you catch every single time they're there, and you catch the same individuals over and over and over again to the extent that you feel like you probably know everybody in the population. I fell in love with kangaroos at like that minute. I was just like, these guys are pretty awesome. Yeah. When they kicked the sand at me, I was like, yeah, you're an interesting little creature. I like you. They're also just really cute. Yeah. They grow on you pretty quick. Whereas like that Microtus in that picture, like I never learned to love meadow voles. They are not that cool. They're just not. They bite, they spray urine in a fountain at you. Like every time you handle them, you get covered in their excrement. They're just not that, and they're not that cute. Kangaroo rats are cute. They are just owl food. Yeah, these guys are special. Okay, so a really simple method that we can use to estimate populations is the Lincoln-Peterson estimator. And there are lots of these really simple formulas. Generally, we use much more complicated computer-based modeling now to estimate populations. But even our really complicated computer models that try to take a lot of variables into play when they make estimates still have some component of this basic formula. So our population estimate, big N, is a function of M, the number of individuals you captured, marked, and released into the population as a whole, times the total number of individuals you captured in a sampling, over the number of individuals that were recaptures from a previous marking event. Okay, the +1 here is an adjustment for small population size so that you never end up with a 0. Okay, and so this is basically just the number of individuals that you captured, marked, and released in this session times the total number of individuals you captured divided by how many of those were recaptures from the previous session. And so it's a mathematical estimation that relies heavily on your ability to successfully mark and release individuals and have a greater proportion of the population marked over time. Little n is the n you count, and big N is the estimate you're after, your attempt to find the total truth of the population. Yep, so they're actually different variables. It's just the lowercase versus the capital. No, they're both number. So it's the number of individuals you have in your hand. I've got this many. I can count them in my basket versus how many I think are in the population based on my estimate that's intangible, which is big N. So little n is truth that I have my minimum number in front of me, and big N is my estimate of how many I think are in this population. Okay, so in order for Lincoln-Peterson to work really well, we have to have closed populations, which means that in this sampling period, there is no change in the population size. So that means that you have demographic closure. No individuals are born or died during the sampling period. And geographic closure, nobody immigrates or emigrates during that sampling period. That's pretty impossible to do over any long period of time, right? And so our current population estimate models allow us to assume closed populations during a short window, like a 1 to 3 day window. And then you can allow the population to be open between trapping sessions before you go back again. So you do one round of capture and marking, and it's closed during that time period while you're doing that event. And then the population is open for a week, a month, a year until you go back and sample again. And you can accommodate that in complex computer models for population size. Okay. Other important assumptions is that all animals are equally likely to be captured. Each sample is random. You're catching a random assortment of the animals in the true population. Marks are not lost between sampling, so nobody ceases to look like how you identify them. And all marks are recorded. The observers are perfect and always detect marks. Okay, these are also tall orders. You guys are going to see that today when you guys look at some salamanders and you can't tell what their mark is. You're going to be like, oh, I thought that should be pretty straightforward. It's not always super straightforward. So basically, the mark recapture method is to go out and capture as many individuals as possible, mark them and release them back into the population, and then go back at a later time and capture them again. And so what we're going to do today is visit some plots that have been in existence since the spring of 2016. And animals have been captured on those plots regularly every spring and every fall since then. And so many individuals are marked. So, you know, about 6 times, 7 times, 8 times a year, we've gone out to these plots and marked and released animals. And so some decent proportion of the population has been marked in the past. We will likely also catch some animals that have never been marked before and mark them today and release them and add them to our marked portion of the population. Okay. Did somebody have a question? Oh, what's your question? We're going to give them a tattoo. Yeah. Um, what does that look for? You've tattooed lots of salamanders. You'll be well. Only those people who have tattooed salamanders before are going to tattoo them today. You and me and Taylor are going to be the tattooers. I don't have any grapes today. Sorry. You have grapes? Kate was going to bring some. If Kate shows up, she might have grapes. You can mark something. I'll let you guys mark plastic bags or something so you can experiment with the dye method. We'll look at some different ways that you mark animals, and I'll show you guys how we mark salamanders. Armando, did you have a question? Right. So like everybody's likely to be captured within your sampling area. And so basically what it's saying is that like you're not better at avoiding capture than I am. Like if we're all in this room, we're equally likely to be captured. Right. Right, depending on the animal size, right? Because if I'm capturing bison, I might not actually be like capturing them, shooting them with a tranquilizer gun and getting them down on the ground. I might be flying over the valley in a plane, looking down with my eyes or with a camera or maybe with lidar where I'm actually looking for the heat signatures coming off of the animals. And so there's lots of ways to capture and your ability to evade capture depends on the method. Yeah, right, right. And with the salamanders, we're only going to capture those animals that are at the surface right now because they can evade capture by going down in the soil. Yeah. So I just have some examples of ways that we can capture animals. I spent most of my population ecology life capturing animals like this. So this is a Sherman trap. It's a little metal box, and it's like a Havahart. If any of you have used a Havahart, you put the door down, and the door has a little treadle that holds it in place. The animal goes in, it hits the treadle, and the door closes behind it, and it's trapped in the box. And so then you can go back and pick up the box and take them out of the trap, right? Oh yeah. Yeah, we would capture everything that we can. Yep. And then but you write that down. You record whether they're males or females or juveniles or adults, and then you put those values into your modeling. Yeah. Today we're going to capture salamanders using this method. So basically we've put out little boards, wooden boards on the surface of the soil to act as natural hiding objects, natural cover objects that the salamanders might go underneath when they're at the surface. And so they're not actually trapped in a box. We're going to lift up the board, and if there's a salamander there, we're going to try to grab it. And so then we're going to capture it with our hands. You can't see very well in this picture, but this bear has a tranquilizer dart right here in its hind leg. It has been tranquilized with a tranquilizer gun. The bear will fall asleep. They will catch it as it falls out of the tree and mark it, move it, put a radio collar on it, whatever you need to do. Huh? Yeah, use a blanket and then you move in. You don't use a trampoline because that makes for those terrible videos of wildlife managers bouncing bears off of trampolines. That's a YouTube video not to go down. More than one person has tried to catch a bear with a trampoline and like, yeah, not smart. One can hope. They're pretty durable, but did you have a question? I don't know. Oh yeah. So this individual here is capturing a lizard with a lizard noose. Nooses are a really common way to capture lizards and snakes. It's a little elastic cord on the end of a stick, and like the lizard sits there and it's like, you can't see me, you can't see me, right? And so you slip it around their neck and pull the cord, and then you can pick them up without them getting away. And then this is how we captured bighorn sheep. This guy is jumping out of a helicopter onto this animal. You can directly jump onto the animal and basically wrestle them to the ground, or you can shoot a net gun over them. So for the bighorn sheep, we used a net gun, and basically you have some wiry little New Zealander riding on the rails of the helicopter with this big rifle with this net mounted on the end of it. And the net has lead weights around the outer edge of it. When you get over the animal, you shoot the net over them like something out of a cartoon. Um, and the net goes over them and they kind of tumble a little bit in the net, and then you can jump on them and basically tie their feet together like a rodeo cowboy. Um, so you might think, and like as I say this, I know it sounds super cowboy and it sounds like this is crazy, like people jump out of helicopters onto animals. But things like bighorn sheep and white-tailed deer are really sensitive, and they die really easily in capture and handling, and they succumb to heat stress really quickly if you pack them into a small area, and they have unpredictable responses to tranquilizer drugs. And so when you have animals that have consistently unpredictable responses to tranquilizer guns, you lose a lot of individuals to tranquilizing, just like some people have problems with anesthesia and can have a bad reaction to anesthesia. And so it has been found for many large ungulates that actually the jumping out of a helicopter or net gunning is the lowest mortality way to capture and handle them. But you want to handle them as rarely as possible. So you try to handle them once, maybe put a radio collar on them or mark them in some way, and you try to not handle them again for 5 years or something if you can. You try not to handle them any more often than necessary. And if they have a way to individually recognize them without capturing them, then sometimes you can just see them and know who they are and not have to ever capture them to mark them or identify them. Okay, so we capture animals and then we mark them. And so as I was just saying, like things like zebras, eastern spotted salamanders, organisms that have unique coat patterns or unique body patterns are individually identifiable without giving them any additional mark. Some of that can be done just like you look at them and you can tell them apart. Other individuals now, especially with facial recognition software and all the advances made in that, you can take a photograph of the animal and then know it. This is even true for passive monitoring methods like trail cameras. Trail camera gets a good side shot of a bobcat, and you can tell that bobcat apart from other individuals based on its coat pattern repeatedly. Same with like cheetahs and anything that has like a unique coat pattern. You don't ever have to actually mark them in order to tell them apart. This is me again marking a kangaroo rat, and the way I'm marking this kangaroo rat is with a PIT tag, a passive integrative transponder. It's the same thing that you might put in your dog or cat if you have your animal microchipped. It's that little grain of rice transponder. You inject it and put it in subcutaneously. So you put it in the space between the skin and the muscle layer, and then you allow that space to heal, and then you scan them with a barcode reader, basically, to tell who they are. These individuals are my sheep. I gave all of these sheep the world's most ridiculous jewelry. They all have these big ear tags, which have a color and symbol pattern on them, so that I could identify them uniquely through a spotting scope at a distance. And so I did that to those guys. I feel kind of bad. They're also wearing radio collars. This chipmunk has an ear tag, a color flag, and a radio collar. That's a really decked out chipmunk. Try to do as little of like we try to not overmark animals at this point. We're moving into a time of trying to have the least invasive marks as possible. That feels a little invasive to me on that chipmunk. And then this is a salamander that's been marked with the visible implant elastomers that we're going to use today. It's an inert elastomer. It's a two-part elastomer that solidifies at cold temperatures. And so you inject it using a 31 gauge tiny diabetic needle, again, subcutaneously in the layer between the loose skin and the body itself. And then you allow that to harden under the skin, and the elastomer glows in the dark. And so we'll have individuals that are marked with little color spots of elastomer. And so we have 4 colors of elastomer and 4 points in the animal's body, the 2 armpits and the 2 where the 2 legs come in. And so we can make a unique combination of colors in order to give that individual a unique name. Okay, so this is just a recap of this slide. Capture as many as possible, mark and release, and go back and capture again at a later time. It's hard to meet the criteria of the assumptions of the Lincoln-Peterson model. This says that actual population, or this is the fact that we always violate the fact that a population is closed because populations change, right? Animals are born and die. Animals move between populations. These are kangaroo rat pups that were born in one of my traps. And so I went to take the animal out of the trap in the morning, and I had mom and 2 babies. I put these near the burrow she headed to. I hope they survived, but I don't know, right? And so we know that birth happened during that night that the population was supposed to be closed because the babies were born in the trap. You also have changes in response to the environmental factors. So if you're trying to catch nocturnal animals, you have to be out at night. You can't try to catch them during the day, right? That's really easy. But also animals respond a lot to weather. kangaroo rats are particularly sensitive to moonlight. They don't go out on full moon nights because they're more easily detected by their predators, and they lay low during the full moon, and they prefer to be out when it's darker. So temperature, weather, snow, wind, cloud cover, all of those things can impact how active individuals are, and then that affects how likely you are to capture them. You also have differential responses to traps. I told you about those desert kangaroo rats that don't like to go in traps, and I would never catch them. But some individuals are also what we call trap happy. So trap shy means they don't go into traps very easily. Trap happy means they love traps. This is a kangaroo rat that we affectionately called Stumpy because he had a damage to his tail, and his tail was kind of short. Stumpy loved to get captured. If I was running traps, they're like in a 10 by 8 grid, and I caught him in row A and I'd let him go. Like as I'm walking down row B, I'd hear a trap close. I get to Stumpy again in row B. I let him go and I hear a trap close. He's over in D now. And like he would just work his way across the open traps, getting as much free food as possible all night long. And so I would like not just catch him every time I had traps open, but I could catch him multiple times in the same night. And he would just be, we are, okay, okay, go here, has your food. So some individuals are really like they get into it. They're like, if I get captured, I get this free meal. I know what's going to happen. And other individuals are like, there's no way I'm going into that scary box. And so you have this differential trap success. You also see differences between sexes and dominant and subordinates. So like coyotes will learn where camera traps are, and alpha individuals will avoid the camera traps, whereas subordinate individuals will get caught on a more regular basis. There's all sorts of interesting dynamics that can play out in the population that you can't necessarily predict as the person that's trying to catch animals. You would think that Stumpy, who had a damaged tail, might not want to go in traps if that tail damage had come from being in a trap, but it didn't seem to slow him down. But if an individual has a bad experience in a trap, they might then avoid it for the future because they had a bad experience. Okay, then there's efficiency of the traps or the searching method. How likely are you to actually record the animals that are present? There are efficiency of the traps, the efficiency of this and skill of the observer at finding animals or successfully removing them from traps or detecting them in a remote sensing like a camera or a drone flyover. And then just to come back, the Lincoln-Peterson, we can assume that the population is closed in short windows of time and allow it to be open during longer windows of time, which is what we're going to do for this study. So I want to really quickly just introduce you to the plots, and then we're going to go outside. So some of you have seen these plots before, or maybe have encountered them and didn't know what you were stumbling onto in the GCC forest. We have 6 plots, and you can barely see here, but there's a row of boards here, a row of boards here, a row of boards here, and a row of boards here. And then there's this PVC tube in the center. The PVC tube contains data loggers that record the temperature once every 4 hours, 24 hours a day, 365 days a year. And they're buried in the tubes at the surface, so at 0 centimeters depth, at 10 centimeters, 20, 30, 40, and 50 down in the soil. So they're recording year-round the temperature of the soil at those different depths. And then we go out and check and look for salamanders under these plots on certain days and can correlate our trap success with the temperatures. And we can look at overwinter survival based on how deep down did the soil freeze. If we have a deep freeze, a low freeze, we would expect that we'd have greater overwinter mortality than if we had a shallower freeze that the animals were able to escape from by digging down in the soil. We're going to lift up the cover board and look and see if there are any salamanders. If there are, we're going to capture them and put them in a Ziploc bag. And we're going to bring them back to the classroom where we're going to measure them and check and see if they're already marked. If they're not marked, we're going to give them a mark, and then we're going to take them back to the forest and release them. So this individual is being measured with a ruler. And then somebody was asking about grapes. We use grapes to simulate salamander bodies because a wilty grape is a lot like a salamander body, to practice giving marks so that before you ever actually give a tattoo to a salamander, you've practiced a little bit. And we also are going to record some physical characteristics of the environment. We're going to record the temperature and the cloud cover and what the weather is like. whether it rained in the last 24 hours. And then we're going to take a soil sample, and the soil sample will allow us to determine what the soil moisture is at the surface. And that can be a good indicator of whether or not you're going to have high captures of salamanders. So we'll take a soil sample, we'll bake it in an oven and dry it, and then measure how much water was lost. And then this is the data sheet we're going to use. So we're going to fill out this part while we're in the field. We're going to write down the date, what plot we're visiting, a modification of the Beaufort scale for wind, who the observers are, start time and end time, air temperature, soil characteristics, whether or not it rained, and whether it's cloudy, overcast, or sunny. And then we'll bring the animals back and we'll record these data. We're going to make sure that we always record the cover board that we caught the animal under and the plot that we caught the animal at on the animal's bag itself. So what I'm going to have you guys do, let me go ahead and just stop this really quick.